No, I think it's that. But yeah, we have two balance. Okay, that's going to work. And we're starting because we have the hour on half the hour. I would start to say welcome, dear pharma colleagues, and I give you an opportunity to get a little insight in what we're doing at Therapeutics. What we're doing these 2 days, we are doing an R&D review to go over our programs, our processes, and future science. And then with that, when we have something really good, and that is, to say, tuning for the future, you also want to work with the people that represent the future and the global knowledge. And that's why we've been attracted in a number of clinical and preclinical key opinion leaders into this process. And I think that's a validation of what the company has done over the years, that everyone we ask say, yes, can I join? And with that spirit, we get a little bit of vitamin injection. Now when we also go through and validate, check, criticize, and do, to say, the next step for the company. And that is an evolution of all the things that we've spent over the years with Therapeutics. So for you to say, what we call outside Therapeutics in the pharma environment, for you to get an insight or an objective view what this siRNA means in the broader context science, and in our case, with optimal. And for that reason, we have invited one of the leader of siRNA, Professor Klaus Wahlstedt from University of Miami. That is including the Bascom-Palmer Eye Institute, which is the #1 eye institute in the US, to say the world. He will give his perspective and a short introduction, and Klaus will talk a little bit more, introduce himself. But a medical doctor with background in forming the Genome Institute at the Karolinska Institute in Stockholm. Then also taking the Scripps Institute to Florida, and now being at the University of Miami. And with the broad network and knowledge in RNA, and I'm happy that we've been able to attract Klaus to our work. Please.
Thank you. Thank you very, very much. So I'll stand here with 2 screens so you can. So thank you so much for the kind invitation here, Andreas and Jose Maria and everybody else. Really, we started this 2-day session today, and it's been really stimulating, and we'll keep on going. And so again, this, I know some of you are from PharmaMar, of course, more interested probably in small molecule cancer drugs, which I have an interest in also, actually, but today will be on the RNA side of things, so the oligonucleotide therapeutics primarily. And as Andreas said, you know, I'm at University of Miami now, School of Medicine. So I'm a Dean for Therapeutic Innovation because I worked in pharma industry also a good number of years, so I try to help different people. But we have this medical center in Miami is claimed to be the second biggest in the U.S. After Houston. Houston, as you know, with MD Anderson, has a huge facility, of course. But here we have lots of different things, but the two most profitable for our medical system is of course the cancer center, with all the chemo drugs and everything they administer, and also the Eye Institute, Bascom-Palmer. So they're very active. You know, they bring in a lot of revenue to the medical center, and we can, pursue a number of projects with them. Previously I'm Swedish, like Andreas originally, and I was at the Karolinska Institute. I remember there was an initiative here in Madrid called Genoma España that I was down here many times actually, but so at some point I was interacting with Madrid, but that goes back some years now. So anyway, so we're going to talk about genome-informed therapeutics, and briefly then, again, not so much about chemistry today, but a little bit about chemistry, Jose Maria. But so we're going to talk about the genome and the transcriptome, right? The RNA end of things. Just scientifically, I've long been interested also in, there are two strands of DNA and RNA, and people tend to ignore that second strand, the opposite strand, but it actually is functional in, pretty much all the time, and I'll come back to that a little bit. So that means that, we have a lot of RNAs also that are not coding for proteins. So we call these non-coding RNAs. We've been very interested in that. So this talk is a little bit also about the science I am interested in, and not only about the drugs we try to develop. But so from a more basic science angle, these non-coding RNAs are exciting, and they include what we call antisense transcripts, which are expressed on the opposite strand of the DNA or RNA. That leads us in then to, of course, the oligonucleotide therapeutics, which is why I'm here in the first place, trying to help Sylentis as best I can. So many years ago, we were actually the first group to administer antisense oligonucleotides into the living brain. And now that's become a very popular thing. There are literally hundreds of clinical trials for the brain, and there are even a few now approved drugs with more coming, no doubt. But CNS and ocular, the eye that Sylentis focuses on, there are many similarities, and what we've learned from the brain we can, I'm sure, apply to some degree at least to the eye. And mostly it has to do that we've discussed this morning about reducing, gene expression. So you have a gene that, does something that's not good for a particular disease, and therefore you want to reduce it. Just like you have a small molecule inhibitor for a target in cancer or another indication. What we've been more academically, but also a little bit now, some companies are starting to do is also use similar technology to also upregulate genes. So in cases where you have too little of something, you may want more of it. And what comes to mind in particular then are some genetic diseases, right? Where you could have one bad gene from one parent and one good gene from the other parent, and you now want to, it's called haploinsufficiency. I'll come back to that. That's the so you can correct that imbalance, if you will. The examples I'm going to give that very quickly towards the end one is a type of epilepsy where you're actually lacking a certain sodium channel called SCN1A in the brain. And by increasing that expression, you should be able to treat a form of epilepsy called Dravet syndrome. Another example, super quickly, has to do with the mTOR system, and I'll come back to that, and that's of interest to many people, for example, in the context of aging. But I joined the pharmaceutical industry first for AstraZeneca and then Pharmacia and Pfizer in the 90s, and many of you will remember back then we were hunting for new drug targets, certainly in the genome. We had, various partial approaches at that time. If you were interested in kinases, let's say, or G protein couple receptors, or certain ion channel class, you could, try to clone the unknown ones by homology. This is before we had the human genome. You could focus on express sequence tags. So this goes back to the 1990s. But then, of course, the Human Genome Projects came. Human Genome Project was a publicly funded effort, as I think you all are aware. There was a company also called Celera that Pharmacia, as Andreas will remember very well, that Pharmacia, we were the first to actually fund this company. It's a company run by Craig Venter, and they actually outcompeted, the human genome effort. So in year 2000, this was published in Nature. This was the human genome effort, and this was the Celera effort, and they were more or less published at the same time. So we finally had the human genome at that time, and you may remember President Clinton and many others were making statements about this. However, there were many things lacking still. This is kind of, but sort of last year or so, or in the last year or two, really the gaps have been filled. So we have the whole human genome now in a nice way, and so there are very few parts of the genome now that are not known. So that's about DNA, right? I'm going to move to RNA very soon, which is, of course, what Solentis needs to work on. But genetic diversity, it's still. Just give you one example. We have you may know of the UK Biobank, which is a wonderful resource where they have, taken lots of people in the UK and sequenced their whole genome, literally, hundreds of thousands of individuals had their whole genome scanned. But as you can imagine, if it's in the UK, people are pretty homogeneous in terms of ethnic diversity. So just one example, there's a biobank actually set up in Miami called Galatea, a pretty new company. And if they, do the same type of analysis and now use, people from different ethnicities, you see there's going to be much less blue. So they have much more diversity, and they're still working on. And pharma, is to some degree at least concerned about this ethnic diversity going forward. So you're going to see probably more of this type of effort in the future. But that's our DNA. So let's move over to RNA. And this is then the effort that I've been involved with also since many years. It's called phantom or functional annotation of the transcriptome of mammals. And there were, I think, some participants from Spain too, early on at least. And this effort then, and this is primarily funded out of Japan with the Riken Institute, their kind of NIH equivalent. So long story short, it started out again in the 1990s. We met in many times, of course, and worked up, the transcriptome to greater detail. Needless to say, learning about the RNA is much more complicated than learning about DNA. So DNA, you can just take any cell and measure your DNA. For RNA, you need to sample lots of different tissues and cell types before you have what we call the transcriptome, as Tamara talked about this morning also. So, but basically what happened here, at this point, people thought there were about 100,000 human genes. Turns out there are just a little over 20,000 human genes, but they're very much more complex than we had thought. And so this went through a number of stages here. A really interesting stage was about 20 years ago now that we realized, that almost the majority of the human genome is actually transcribed into RNA. So as you may or may not know, I mean, just 1 to 2% of our genome corresponds to protein coding genes, but more than 70 or 80% of the genome is actually being used, if you will. It's being transcribed into RNA. So all of that additional RNA is what we call non-coding RNA or non-protein coding RNA. And just so that was kind of a real insight, I think, based on this project. And then there was a project funded by the NIH called ENCODE that kind of a few years later concluded exactly the same thing, that more than 70–80% of the genome is actually transcribed. So there's a lot of, the bottom line here for a company like Sylentis is that there's a lot of RNA, that can be targeted with their technology. So that the number of drug targets for Sylentis is just infinite. There's so many, and so that's of course good news, because there's not going to be a target shortage. The issue is to pick the right ones, because clinically, of course, you can only pursue so many. But at the front end, there's so many possibilities that, of course, we couldn't have dreamed about before this type of effort. And this is still going on. This effort is focusing on what we call enhancers that are very important in cancer. I'm sure the pharma drugs are affecting enhancers in some way, and promoters. I think you mentioned also, Maria. These are regions of the genes, right? These transcription factors you mentioned to me last night are, I'm sure, affected by these type of mechanisms, and the pharma drugs are in some way tickling these regions, and of course, something that's very fascinating. So going on here, I'm not going to keep you forever. So transcriptomics, Tamara just mentioned. It's something that is much easier to do today, of course, compared to 20 years ago. There's much new technology. There are basically two ways to do it. The standard sort of RNA-seq protocols. Primarily a company called Illumina, that's, I'm sure is what Sylentis has been using. But there is also, other ways of doing it. These are kind of modern arrays. We talked about Affymetrix this morning. They don't exist anymore, but they left behind these very complex microarrays that can still be used, and we still use them. And they're actually a little better to see these non-coding RNA transcripts because we're going to miss a lot of those by these standard RNA-seq protocols. But this is kind of technical, so I'll skip over it. But we wrote a review on this quite recently, if anybody's interested. So at a much more fundamental level, biologically, right? You all heard about the central dogma. Formulated in 1958 by Francis Crick, arguably the greatest scientist of the past century, at least in biology and medicine. So Francis Crick was, of course, famous for, co-discovering the DNA helix back in early 1950s. But in 1958, he had enough evidence to suggest that central dogma of all biology, right? That DNA is transcribed to mRNA and which is then translated into proteins. So this is a central dogma. And actually, Sidney Brenner, who had discovered mRNA only in around 1961, so it's pretty amazing that Crick was able to postulate this without really having detailed knowledge of mRNA. Sidney Brenner, and of course, they both got the Nobel Prize. Sidney Brenner, I was at the Karolinska and was actually hosting Sidney Brenner in 2002 when he got his Nobel Prize. So very fascinating man. But these non-coding RNAs, what we suggested a number of years ago, and I think it's held up pretty well, is that non-coding RNAs can then regulate this central dogma in various ways. So microRNAs that were mentioned this morning will primarily work at a transcriptional level by affecting mRNA stability, whereas many of the other non-coding RNAs, these novel things in the genome, can act also at a transcriptional level, so to hold back or stimulate, the transcriptional process from DNA to transcriptomics to mRNA. And all of this, you know, Sid Brenner was very fascinated by this discussion, certainly. Crick died in 2004, so he didn't really have a chance to discover this new type of thinking, I think. Anyway, so in terms of this very complex, you know, transcriptomes, the RNAs, right, in higher organism, this is the way we've been thinking about it, and if you look here in the red little dots here, they are, each of those represent a different species of, and this is in terms of protein coding genes. So let's say an ion channel or a kinase or something, you know, something that maybe where PharmaMar's drug work, they would be more in this category. And the point of the slide here is that, you know, they kind of level off with evolution. So many of them, many of these proteins are kind of similar if you are, you know, a mouse, let's say, compared to a human. With the blues, on the other hand, you know, you start here at the very simple organism called mycoplasma. You have virtually no, you know, no non-coding RNA at all. All of it is protein coding. And you then move up to higher organisms. So these are organisms that are just one cell, right? And then 3 to 8 cells and so forth, and up to humans, which we have about, you know, almost 200 different cell types. And in terms of this non-coding RNAs, it's still a hypothesis. It cannot be proven probably for many years, but we think that, you know, what has driven evolution is a lot of these non-coding RNAs that are then more complex. So here we even find differences between humans and non-human primates. So a lot to talk about here, but I think you get the drift that non-coding RNAs perhaps is a key driver of evolution from you know, simpler organisms to more complex organisms. And again, this opposite strand has been kind of ignored, right? This is Jim Watson, of course, who's the co-discoverer, of course, of DNA, who's also fascinated by this space. This is you know, very detailed, but just from a recent review we wrote that these antisense transcripts can look, and I'm going to skip over this very quickly. They can be found in the genome in many different ways. So I'm not going to go through this today, but you know, this is what bioinformaticians do for a living. They really try to go into the genome and try to find what different. But all of this is, much of it is still unknown, and it's very hard to find these sequences sometimes. And we've kind of been specializing in that. And there are then multiple ways that, you know, these antisense transcripts can work. For example, by interfering with the coding gene and transcriptional interference, you can form a DNA RNA triplex, and there are also other mechanisms. I'll skip over this quickly. And you can affect splicing in this by these types of mechanisms. You can affect polyadenylation, methylation, certainly of RNA. Methylation doesn't only occur at DNA, but it also occurs at RNA, and other modifications of RNA. So very complex and kind of evolving field. But back to maybe more bread and butter here. So the oligonucleotide therapeutics, and here I'm mainly going to focus on ASO, or antisense oligonucleotides, or siRNA that you've heard a lot about before from Sylentis, I'm sure. I'm not going to talk about CRISPR, although you use actually oligonucleotides also for CRISPR and gene editing type of approaches. So primarily you do this to you know, knock down a coding gene. With antisense oligonucleotides in particular, you can modulate splicing. So you can see that a certain exon is preferred over another exon. It's called exon skipping. It's harder to do with siRNA because, but there are tricks maybe also for siRNA. And as I said, we can also not only reduce a gene of interest, but you can also upregulate to get more of a gene of interest. All in all, of course, this technology is used for undruggable targets, as we say, where a small molecule is not really feasible. And also an antibody cannot really do the trick. So, an antibody can usually work when the target is somewhere out of the cell or in the cell surface. So ideally, you'd want to use this technology for targets that are inside the cell, right? And also not, druggable with small molecules. So like, if you gave me a kinase or a G protein coupled receptor or an ion channel, I would first certainly advise people to try to find a small molecule. But if there are, other, we talked about kind of a scaffold protein this morning, maybe in the Wnt pathway, right? Not great siRNA target, I think, because there is really no way I can see to make a small molecule for that. So that would be a very good example. But the point is really any RNA, right? And again, like the point I already made, there are endless numbers of RNA that you could theoretically target if you had this technology really nailed down. So my little journey here is started again also a long time ago in the 1990s. So we were the first team to actually apply oligonucleotides to the brain. This is in this case a mouse here, and we're giving it, it's called intrathecally here. So this is, so you get inside the blood-brain barrier, and you can also give it ICV or intracerebroventricularly. But both cases, you do this because these oligonucleotides do not pass the blood-brain barrier. Really zero. They're so charged, these molecules chemically, right, that they're not going to do that. And with the eye, you partly have the same issues, but you have some advantages, of course, over the brain. But there's certainly some similarities too. So, but what's kind of nice here is that this work. Initially very few companies took notice. It probably took more than 10 years before any companies really started working on this. But then once they did so, and now this is a human doing the very same thing we did in mice. You would inject here, intrathecally. So this is to get, again, access to the fluid, right, the cerebrospinal fluid surrounding the brain. And there are, these two recent approvals. This is for Spinraza, is a drug from Biogen for spinal muscular atrophy, which is a really bad, childhood disease. And so this was just approved, and even more recent approval is this drug called Sodi for ALS, another really terrible disease. So both of these are then in patients given intrathecally. Just a little side note here in terms of, we had this discussion this morning also about approval processes. Actually, in terms of that latter drug, the ALS drug here, actually the data wasn't very good at the time. Now it's getting better that it actually helps patients. So the approval was more based on the fact that a certain biomarker called neurofilament light was reduced. So the significant reduction of a biomarker was enough for the FDA to say that we will approve this drug. So I think it's an important point to make, that, in some diseases, there's such a big need for a new drug. And specifically, this targeted a gene called SOD1, which is a subgroup of ALS. So it's a small group of patients with ALS, about 300-400 patients in the US. That's about it, right? And globally, a little more than that, of course. But that's still enough, you know, for kind of a very successful drug in this space. A company then that I'm sure at least the Solentis people here are very well familiar with is called Ionis. So they took this technology, and they focus on antisense oligonucleotides. And I think you can see these are the various CNS disorders that they are pursuing. So a number of, there's certainly other companies doing this too, but just to give you a flavor. So they're working on diseases like, you know, Alzheimer's and Parkinson's and ALS and Huntington and so forth. So a number of different disorders here. The technology is okay, I think. You know, it allows you to give this intrathecal injections once every few months, which is I think doable for some patients at least. The degree of knockdown, you know, the efficacy, if you will, is okay. It's you know, can probably be improved with newer technologies, we think. There are some question marks. You know, the doses you give intrathecally are pretty high, and also, you know, you do build up some immune issues, you know, by repeated injection of these drugs, and it's still being evaluated. So there are things to be certainly worked out. Then, of course, RNA interference, you know, came along 2006. I was still at the Karolinska, and I was very much promoting this prize, of course. We had an RNA Nobel conference that same year preceding, where Fire and Mello came, and they got the Nobel Prize the same year. And you know, but initially, as we also discussed this morning, you know, as a therapeutic RNAi drugs, siRNA drugs for brain and eye was kind of disappointing, but there are some certainly improvements with recent, you know, modifications and so forth. This is way too small to look in the back, but this is just to state, this is from a recent review from some colleagues at Alnylam, actually, but just saying that, you know, there are different places. So the antisense oligonucleotides will work through what we call an RNase H mechanism, which is inside the nucleus. Splicing, you know, is a correcting splicing is very commonly done, also typically with antisense oligonucleotide drugs. And of course, the siRNA type of drugs will work through this RISC complex you've heard about here, I'm sure, before, mostly outside the nucleus, but probably to some degree also inside the nucleus. And here are some microRNAs that are, of course, very similarly, where mechanism is quite similar to the siRNAs. So Alnylam is one of the companies pushing siRNAs for the brain and to some degree for the eye. I know Andreas has been talking to them recently here. And just one example, there are many improvements actually done by also smaller companies. Also in China, I'm aware of several companies doing similar work with different types of modifications and conjugations that are all aimed to improve the technology, of course. But just one example here from Alnylam. This is a kind of a fatty chain, C16, that they have just here added to their modified siRNA, and they then applied this in a project for Alzheimer's disease, targeting APP or amyloid precursor protein, and this is now pursued for Alzheimer's and also for, you know, a somewhat related disease called CAA, or cerebral amyloid angiopathy, where too much amyloid is the problem. So the idea here is to reduce amyloid, and with this modification, which incidentally, you know, occurs also in other type of drug. Ozempic, for example, has a C18 chain, so it's nothing totally unique for this drug. So it's a pretty commonly used trick to improve really the distribution of the drug in the brain. But But the nice thing from Enlilum and others here of their disclosed data is that the effect is quite long-lasting. So perhaps opening the door for giving these intrathecal injections as infrequently as twice a year. So if you have these intrathecal injections twice a year, I think that's very feasible clinically for many diseases. Certainly for ALS, as I mentioned, there would be no issue. This drug we discussed, I think last night also, Leqvio from Novartis. It's same idea here. You give the drug, in this case then subcutaneous twice a year, and now with this GalNAc conjugation that I think many of you are very well familiar with. You know, there's a little sugar molecule that kind of tags on to a receptor in the liver cells and then internalizes and stays around for a long time. So also this drug is given only twice a year, as we discussed last night too. A lot of activity today. How do you try to get something into the brain through the blood-brain barrier? And the most common trick is to attach your cargo, in this case an oligonucleotide drug, to a transferrin receptor antibody. This is kind of a Trojan horse type of trick, where you have this antibody that finds, you know, these transferrin receptors on the blood-brain barrier and then internalizes. So there's transcytosis of the whole complex here, including the oligonucleotide. And Roche is one of the companies that has been working very heavily on this. But I would say still the data isn't quite as strong as it needs to be. So they have maybe an effect size of 30 to 40%, which is probably not enough. But at the conference I just attended last week in Boston, there are probably 10 companies working on variants of this, you know, with other types of antibodies and mostly around the transferrin receptor, but to improve this type of technology. So, this is going to be hard for you to see, but I pulled out this. This is ophthalmology now, of course, which is the topic of the reason I'm here in Madrid. These are then, I found 19 different, you know, oligonucleotide projects in ophthalmology. You're going to find Sylentis here in 2 places, of course, as you know. So, so it is a field that is bound to grow. I have no doubt about that. But sorry, that's pretty small. I'm going to skip through very quickly now the rest of the talk, because the rest of the talk, up till now, we've mostly been talking about reducing gene expression. But there are, you know, certainly strategies to also increase protein production. You know, of course, you know about protein replacement therapy, you know about gene therapy, gene editing. You know, there are many companies now working on gene editing with some degree of success, actually, and even approvals, as you've seen here in the last year. mRNA, of course, got very popular with the COVID vaccines, but also mRNA to increase protein there are a number of efforts around that. But then you have to use lipid nanoparticles. So for the RNA targeting approaches, most people do this with naked molecules, so without any delivery tricks as such. There are then various ways to use oligonucleotides to actually increase mRNA abundance, and that can surround mRNA delivery. It can surround, you know, so-called small activating RNAs that work around the promoter region. It can involve these enhancer RNAs, natural antisense transcripts that I'll come back to, and you can affect imprinting, you can affect splicing, you can modulate microRNA activity. So there are a number of ways to do this, depending on what you want to do. Quickly, and I'm just going to skip through, these are some ways that these natural antisense transcripts work. So we have then, worked on various ways to turn gene expression on with these antisense oligonucleotides. This was then, we had spun out a company from Scripps where I was, which then landed with a company called OPCO and now with a company called CAMP4. So we have licensed this to these companies in Boston. But what this slide shows that you can screen a number of these pretty readily and then find good molecules. Quick example here has to do with a type of disease called Dravet disease. Again, the concept here is that you need to increase the number of these sodium channels. And it's a nice and interesting and feasible therapeutic project because this protein is really not expressed until the baby is between 6 months and 1 year of age. So you actually have a window here to completely treat these patients and maybe cure these patients even. So it's a good example of that. I don't know. So this is an example of one of these patients from a colleague of mine in Miami. So these babies or young children, they can have hundreds of seizures a day, and often they will not develop normally. So it's a very severe disease. As you can imagine here, just skipping through pretty quickly, we found the mechanism here that we can increase the expression of this sodium channel. And what we do here, Tamara, is we typically do this walking around, very careful walking around these so-called hotspots to find the very best oligos to do the job. And then these are we use patient cells in this case, and we can very dramatically, upregulate this particular gene. This also these are the African green monkeys we mentioned this morning also that we have worked on and off over the years. So again, if you inject your drug here intrathecally, you of course wonder, can it really reach up here to the brain, right? To the forebrain where it needs to work? And yes, the answer is yes, it can reach up there. And this is then, we then had a very nice mouse model for this disease because it's very highly conserved between mouse and human. And these are then heterozygous mice. So again, homozygous die, if you knock out this sodium channel completely, they cannot live. That's not compatible with life. But heterozygous will be like these humans that are haploinsufficient. And same story here, these mice get these seizures that are very severe, and so it's a very good mouse model. Really, it's rare that a mouse model will replicate a human disease very well, but in this case it does. So, and I'm just showing you here that we're able to kind of correct this problem with treatment, once weekly treatment with oligonucleotide in this case. So this is now, pursued by a company called CAMP4. In parallel with this, there's another company in Boston actually called Stoke that has another approach to upregulate this gene. They are eliminating what's called a poison exon. So this has to do with nonsense mediated decay. So they are, some of your protein production, is inefficient. So what they're doing is to make more of excluding this, what they call poison exon, that just makes the protein less effective. So it's an alternative approach for this disease. And they very recently published data here that they have some clinical efficacy, which is nice. However, at a very high dose. So an idea a couple of my graduate students had was actually to combine these two methods. So we have this method and the other method with poison exon exclusion. And this synergy was actually quite interesting to see. So this is, again, our approach now with a company called Camphor in Boston. And another Boston company then has this other approach. And you see they both work. These are the red bars here. So ours works a little better, maybe we could argue, but in this particular system at least. But the point is, if you put them together, you get an even much better effect. So right now we're working with both companies actually to try to get molecules to that are even better, that combine these two effects. And to get a single molecule that has both of these activities. Very last point here has to do with a drug, just another example that you may have heard about called rapamycin, certainly well known in the cancer field. But it's also, the one compound that people can agree on actually may extend lifespan of animals and possibly also humans. So a lot of people actually take rapamycin to live longer, if you will. And rapamycin is, of course, this complicated molecule at Stuart Schreiber's lab. Actually, he solved that structure, and I've talked to him about that a number of times. But issue with rapamycin and these mTOR drugs, rapalogs, is that they actually inhibit two complexes, mTORC1 and mTORC2. So, interestingly enough, it's, and this is my last slide I think I'm showing. So mTORC1 is quite famous for extending lifespan. So these are mice, and if you give even kind of a middle-aged mouse rapamycin, that will extend its lifespan. And that has been publicized a lot. What people haven't seen is that this other mTOR complex actually reduces lifespan. So rapamycin will probably be a good thing here, but it will be a bad thing here. So one objective we have with this RNA technology we're using is actually we found some molecule also that with siRNA technology will inhibit mTORC1 and activate mTORC2. And we think for lifespan applications and for liver health, because we can deliver these nicely to liver, of course, this may be a nice project for the future, at least, and we have funding to do that. So to summarize here, I've talked to you about RNA, right, being really, really complicated, more and more complex. But of course, the good news, again, that companies like Sylentis will have an infinite number of targets to consider. I've told you that, both strands of the DNA or RNA actually matter. I have, again, said that, there are many different RNA mechanisms in the eye and in other organs that can be exploited therapeutically. I think we have only scratched the surface of that. There's really so many things that can be done. We can achieve both down and up regulation. Down regulation is more straightforward and will certainly work better at this point, but up regulation can be considered. There has, of course, been, delivery has been the main obstacle, but there is recent progress with different formulations, with different conjugations, and so it's a very, very active field. And I think not so many people are using all this new knowledge for ophthalmology. So I think Sylentis, again, is in a good spot to really become a leader in this space. And again, brain and eye applications are now feasible. if you asked me 20 years ago, I would have been much less optimistic, and even 10 years ago, I would have been less optimistic. So, and this is just some of my colleagues. I'll stop there. Andreas, thank you very much. Mm-hmm. Sure, sure.
What we see here now, we say during 20 years,
Yeah.
that there was aspiration and then more understanding, and now we have something. And you talked about a number of RNA.
Yeah.
Is the abundance of RNA opportunities going to be hard to then conclude something that leads to drug project, because you can be so immersed in different RNAs?
No.
So how do you deal with that?
Yeah, no, again, the bottleneck has really been on getting the right technology going and you know, solving the delivery trick, which, you know, and if focusing on Sylentis, if Sylentis really has the best way of delivering these types of drugs to the eye, then, you know, any RNA is in play, right? So once you have that technology under wraps, then you can certainly consider all of these new RNA targets down the line. Some of them are much less proven, right? And, you know, one thing if I didn't stress it enough, but many of these non-coding RNAs, unfortunately, are not so well conserved. So, you know, so you cannot, usually it works from human to monkey. But if you move from human to mouse or, you know, lower organisms, the molecules are very often different. We cannot make molecules that work across species, which makes it necessary, you know, for development, you have to use a different molecules as you validate your target than the clinical molecules. That presents a dilemma. So I was impressed by, I guess, Tamara's statement this morning, right? That your molecules here, right, you actually made a point out of, you know, them working in different species. Many others, I think, reason that, you know, if you find an optimal human molecule, it's okay that we make another molecule for mouse. But if you have the same molecule, you know, that's even better, of course, because then you can compare apples to apples, if you will, right? So that's a nice thing. But for these other RNAs I talked about that are, you know, we call non-coding, chances of finding one that works in both human and mouse is very small. We've done it maybe once or twice, but usually not. So I hope that answers your question to some degree. Right. Correct.
The other thing is that when we talk about RNAs, knowing more the different kind of diseases related to ophthalmology, you have a toolbox to address different silencing for different diseases.
Correct.
Yeah.
Correct. And also genetic disorders. You know, genetic disorders are often, in all the gene therapy trials using typically adeno-associated virus for gene therapy of eye diseases are cases where you want to increase something, right? So, and that could be cases where you want to affect splicing, right? So there are, and those are not readily, you know, addressed with the siRNA technology we talked about this morning. And so then you may want to add to your toolbox, right? Just like I know you're thinking about doing so. But I guess the good news for Sylentis is that there are a number of eye diseases where we have big medical need. Some of them are more genetic, you know, where we may use some of the tricks I talked about here. Some of them are, you know, more common diseases where you know, but typically you have a single target that can be targeted.
But the thing that
yeah.
With RNA,
yeah.
sit down and look at different eye diseases disease and then see how do I address that eye disease. So
Yeah.
it's a technology that spans over the whole spectrum of eye disease. And to say it's not just that we have a glaucoma project or we have a dry eye. We can go back and forth and see what we address.
Absolutely, absolutely. And ranging from more rare diseases to more common diseases. And that's, of course, a strategic decision the company has to make, right? How rare.
When you say the word abundance, you want to make sure that the abundance doesn't make that you end up in a maze of different projects.
Exactly right. Exactly right.
Any questions from the audience? Yes.
Yeah.
Pharmacology for many years have been based in inhibiting signs,
Yes.
in regulators,
Yes.
inhibiting some bad activity that produce a disease.
Yes.
But now it seems very interesting that you can do the opposite. I mean, to increase activity, acting at the DNA, RNA levels on enhancer or promoters.
Correct.
To increase, to upregulate.
Correct.
Something that is needed to treat certain diseases. So it opens a new opportunity.
Exactly. No, I could. That's exactly right. I have nothing to add to that. Really, it provides an opportunity for many diseases. Again,
I was curious about another thing. Which is the target to treat, prion disease that some companies working in that?
Yeah.
Wondering which is the target.
Yeah, and the target is the prion. The very prion protein, and there are, for example, some people who have a genetic form of it and now try to treat themselves before they get sick. Actually, right? So there's a genetic, prion you can get from mad cow disease and things that you cannot really predict. But there are a small number of people who have a genetic problem, that they will get prion disease. And if they can be, treated before they get sick, that's really the dream now, right? So that's a good question. It is a good example of this technology where it's being used. There's a paper in, was it Science, just 2 weeks ago, if you want to look that up, by this group at the Broad Institute that exactly addresses that question. I'll be around so we can talk more informally, so don't worry. Yeah.
But again, thank you for taking the time to join us, and I hope that we see that we're working in a field that is suited for it, just to manage it correctly and deliver drugs. Thank you.
Thank you.